next presenter is Austin Wiesler. Austin is originally from Nebraska, I just found out, and worked on his undergrad here at SDSU. He was helped out on our captive bighorn sheep work here at state, and then more recently has been working with South Dakota Environment Game Fish and Parks, and just started a master's on bighorn sheep at Badlands National Park. He'll be talking about today as his master's project.
Thank you, John. Thank you, everyone, for showing up. Like John said, I'm in my first year of field study. My master's research, we're assessing survival and mortality of bighorn sheep in Badlands National Park. So, just to kind of give a brief introduction and history of bighorn sheep in North America. They were once numbered at 1.5 to 2 million on the onset of the 19th century. Had significant population declines over the last 150 years. As you can see, their range has changed drastically from northern British Columbia all the way down to Mexico to the west coast to eastern edge of North Dakota, South Dakota, and Nebraska. By 1960s, population declined so much that we were almost at 25,000 in North America. And with recent management actions, we've replaced a lot of these once eradicated bighorn sheep populations back into their native ranges throughout much of the western North America. And today we're sitting roughly at around 87,000 as a population number. So there's three main contributing factors to the decline of bighorn sheep populations. One being unregulated hunting. Not so much a big deal anymore, but back in the 1800s, 1900s, definitely had its impact on the bighorn sheep populations. Today, this is still a problem. Habitat loss, fragmentation, whether that's from human interactions with roads and ski resorts and stuff like that, are taking up, cutting through bighorn sheep habitat to suppression of wildfires that provide prime habitat for bighorn sheep. And last but not least, disease, which is probably the most significant cause of decline in bighorn sheep populations. So to touch more up on disease, bighorn sheep are susceptible to a whole host of to diseases. As a biologist once said, sheep are just looking for a way to die, so they're pretty good at it. Pneumonia being the number one cause and the main obstacle for bighorn sheep recovery in North America. There's multiple bacterial pathogens that are thought to be the cause of pneumonia in bighorn sheep, of which the Pasteurella bacteria, the family of that, and Mycoplasma pneumonia, the two primary pneumonia-causing pathogens. With more focus recently, as we just discovered in 2008, Mycoplasma pneumonia with these huge die-offs from pneumonia, with the main focus on the Mycoplasma pneumonia. So with pneumonia in bighorn sheep, initial exposure to naive populations can cause a 30 to 90% all-age die-off. Infected herds can experience little to no lamb recruitment, and this can become just a stagnant population. This can last decades where you're not getting lamb recruitment, so you're just having sheep populations that are getting older and older, shedding this pathogen and just slowly dying out. Primary vectors for these pathogens are domestic sheep and goats. So as a manager, you want to, our primary management action to prevent pneumonia outbreaks is to keep separation between domestic sheep and goats and bighorn sheep populations. So Badlands National Park history with bighorn sheep. Historically, there was a subspecies of bighorn sheep known as Audubon's bighorn sheep. It was completely eradicated in the 1920s through hunting and a potential exposure to various diseases. The last Audubon sheep was thought to be shot in Jackson County in 1924, right around the Badlands area. And from 1924-25 to 1964, Badlands National Park and the area around actually held no bighorn sheep. So a little history between Badlands National Park restoring the bighorn sheep population. 1964, there's a translocation of 22 bighorn sheep from Pikes Peak, Colorado to Badlands National Park. This population did good initially and grew to about 120 individuals, and then by the 1990s, for the next decade or so, that population slowly began to decrease. And managers, because there was only 22 individuals that started this 120 population, they were a little concerned about population bottlenecking. So in 2004, 23 more Bighorn sheep from Wheeler Peak were brought in to increase genetic diversity and to augment the existing Bighorn sheep population. And as of recently, recent surveys have shown that the population has roughly grown to 175 to 200 individuals in the park. So my project objectives, I've listed 4 here. The first one is to estimate the prevalence of Pasteurella bacteria, Mycoplasma open pneumonia, and to survey for the presence of other potential diseases within the herd. The second objective is to monitor adult Bighorn sheep marked with radiotelemetry collars with mortality chips to identify cause-specific mortality. Third objective, capture and collar lamb Bighorn sheep to determine disease prevalence and cause-specific mortality, as well as accurately estimate yearly recruitment rates into the population. Fourth objective, assess the risk of respiratory disease due to contact with domestic livestock in the immediate area. So to give you an idea of where my study area is, the lightly green shaded area is Badlands National Park. This is just a map showing the northern unit. There's a southern unit of Badlands National Park that actually extends straight down, and the reservation, Ogallala Reservation, runs the bottom of this map. The southern unit, as we call it, is run primarily by the reservation, their natural resource department, so I don't have access to that part of the park. There is a subpopulation down there. They don't really know a whole lot about it, but my primary focus is around the northern unit of the park. And so just to give you an idea of where the sheep are within the park, we've got 4 main subherds in my study area. We've got the Pinnacles area, Pinnacle subherd, we've got the Homestead subherd, Cedar Pass subherd, and then finally the Hayview subherd. To address my first objective, estimating prevalence of disease and to survey for other infectious diseases. In March of 2017, we captured 25 Bighorn sheep using helicopter capture, and they were transported to workup stations where we did complete disease sampling. These sampling kits were designed primarily to detect these pathogens causing pneumonia. We also took blood to test for other infectious diseases. Complete sampling kit includes nasal swabs, throat swabs, blood draw, nasal wash, and weight and conditioning. All our lab work for the pneumonia portion of the project is sent to Dr. Tom Besser's lab in Washington State. Results are then sent back as bacteriology, real-time PCR, and ELISA. So with bacteriology, that's examining a whole host of just normal respiratory bacteria, and then the real-time PCR and ELISA is mainly focused on that Mycoplasma open pneumonia detection. So to address the second objective of monitoring the adult Bighorn sheep within the park, we once again captured that same 25 Bighorn sheep, 20 ewes and 5 rams. Ewes were fitted with a very high frequency collars, and then they were ultrasound to determine pregnancy. If they were, we fitted them with a vaginal implant transmitter to determine lamb partition sites and to capture Bighorn sheep lambs. The rams also, just a little side note, rams were fitted with GPS collars. This was mainly due to game fish and parks' interest in observing mature Bighorn sheep rams outside the park as a way to maybe develop a hunting season around the Badlands National Park area. So monitoring located at least twice weekly. Mortality chips give you an 8-hour period the collar has to sit still in order for a new frequency signal to give off, and that determines that the collar is either dropped or mortality has occurred. Weekly recording of movements to determine where the sheep are using, what kind of habitat, where they're moving around in the park. Third objective, capturing collar Bighorn sheep lambs to determine disease prevalence and cause-specific mortality, as well as accurately estimate yearly recruitment rates. So we started listening for vaginal implant transmitters in our collared ewes in mid-April. Once those bits are dropped, you usually get 48 hours to capture those lambs before they're uncatchable. Lambs were fitted with an expandable VHF collar that expands with them as they grow. These collars typically last anywhere from 3 months to hopefully that first yearly recruitment into the population, so a year, year and a half. With every capture, we recorded weight, sex, and the location of patrician site for the lambs. Monitoring for the first 3 months of life is daily monitoring to get the exact cause-specific mortality if it does occur. Weekly recording of movements to see where parts of the park they're using, where they're moving, and then also yearly movements. These collars we're hoping that will last over a year, so when these bighorn sheep grow to a yearling and they're more prone to expand and look for new sheep populations, we'll be able to track them, see where they're moving inside and outside the park, and yeah, just look at movements. So with that, within the four subherds, 3 of which we caught lambs out of. The Hay Butte subherd is for next year's project or next year's field season. Yes, the red just outlines kind of the sheep habitat where known lambing patrician sites were. And then finally, our 4th objective was to assess the risk of respiratory disease due to contact with domestic livestock in the area. So within 8 kilometers of the park boundaries, we just went around looking for domestic sheep and goats, to determine the operational status, and by that I mean what kind of farm operation they have going, if it's just a hobby where they have a few sheep and goats, or if they have a big operation going where they're bringing new sheep and domestic goats in and out, potentially bringing new pathogens in. And then if the landowner agrees to sample for that pathogen presence within that herd. Eventually, we will conduct risk analysis of disease transmission between domestic herds and bighorn sheep. And this model uses domestic herd allotments and overlaps with bighorn sheep habitat along with telemetry points. So the recording of where the sheep are moving, it overlaps them, and over time it gives a risk of how likely that bighorn sheep population is going to be exposed to that particular allotment of domestic sheep or goat. So just the results to date, disease sampling results. So the top 3, the Manhelium, Rivesteinia, Pastorella, all part of that Pasteurellacea family, and then that bottom one is the Mycoplasma pneumonia, where our main focus was. So of the 25 individuals sampled, 19 individuals cultured some sort of Manhemia species, 3 individuals had Rivesteinia, and 1 individual had Pastorella, whereas Mycoplasma pneumonia, none of the individuals sampled were able to collect a culture off of that. And then within the ELISA and PCR, which was listed for the detection of Mycoplasma pneumonia, we had no PCR detection, so there's no active living mycoplasma being shed from the 25 individuals that we sampled. What was interesting though is we had positive titers from the ELISA test in 13 of the 25 individuals. So at one point these bighorn sheep that we sampled, 13 of them were exposed to this Mycoplasma pneumonia, but they're no longer shedding it. So they somehow got over that initial exposure and are now a fairly healthy population. Another interesting point is these titers were only found in sheep 4 years of age and older, suggesting that there's an exposure to Mycoplasma pneumonia 4 years and greater. So somewhere along the lines of 4 years and up, there was an exposure. Now on to survival and mortality results. Lamb collaring and survival. A total of 23 lambs were collared this last season. Currently there are 15 lambs still collared within the park. Current survival is at 75%. Our sources of mortality or predation, 3 from coyote and 1 from bobcat. Accidental, and by accidental I mean lamb fell into a very deep crevice and starved to death. Unknown, collar went off air and the lamb went missing, no longer with the ewe, so it was a complete unknown death. Abandonment, which is capture related, and drop collar, which is the collar just ripped off. And the bottom 2, abandonment and drop collar, those 2 are censored out of the total survival percentage due to abandonment being capture related and the drop collar. We can no longer track that individual, but it's still alive. Survival and mortality in our adult population. Total of 21 ewes and 5 grams collared. current 18 ewes and 5 rams are still collared. Survival is at 96%. We had 2 deaths related to capture injuries from the helicopter captures, and then 1 unknown death as of recently. So our survival in our adult population is at 96%. Capture-related deaths obviously are censored due to capture. For our analysis for the survival and mortality, we will use a Kaplan-Meier method with staggered entry in program mark. And on to risk of contact. We have located 2 known herds of domestic goats and sheep within 8 kilometers of the park so far. Sampling efforts are actually in the progress right now to test for these disease pathogens within those herds, and heavily surveillance is going on with those domestic herds in respect to the bighorn sheep population. So the landowners are very on board with what we're doing, and they're communicating with us to see if bighorn sheep are coming close to them, and if so, we can take management actions after that. To give you an idea of where these domestic herds are in relationship to the bighorn sheep, there's one down by Interior. One down by Interior is just kind of a hobby farm. They've only got a few goats, whereas the one up closest to the Pinnacles herd, it's more of an operation. They have around 300 domestic goats in that herd, but they're currently selling off a large portion of them. So we will be sampling that herd in the next coming weeks, actually. To give you an idea how close we are to an outbreak if this domestic goat herd does hold some of these pathogens. So you can see here's some bighorn sheep, and in the way back you can see some small white specks, and those are domestic goats. There's only just a highway separating the 2, and they both have been seen butting up close to these fences on either side of these highways. So for the 2018 field season, we'll collar an additional 20 ewes and 20 to 30 lambs during 2018 field season. Expand and include the hay butte subherd into data collection during year 2, and continue mapping of domestic goat and sheep herds around the Badlands National Park. Monitor and document bighorn sheep movements outside the park. Management implications. Provide management to the park to maintain a viable and healthy herd within Badlands National Park. Bighorn sheep are very popular animal to come see in Badlands National Park. Very popular, a lot of tourists come to see them. So maintaining that there and just maintaining the complete ecosystem in Badlands National Park is very important. Also, vital rates from this project can be used for additional research purposes that are going on in other populations in western South Dakota. A lot of disease research is going on in western South Dakota, and because Badlands National Park was a healthy population and a growing population, as we can see, can be used as a baseline for these disease research that's going on in western South Dakota. Also, because bighorn sheep, or the Badlands National Park sheep, is a growing population and eventually will reach carrying capacity, it also is a source herd because it is healthy for translocations and augmentations of bighorn sheep throughout western South Dakota also. Then I'd like to thank partners in funding: Washington State University, Idaho Fish and Game, National Park Service, South Dakota State University, USGS, and South Dakota Game Fish and Parks. I'd also like to thank my advisor, Dr. Jenks, committee members, A. Childers, Dr. Walsh, Dr. Kassir, Dr. Tom Besser, and then South Dakota staff, South Dakota State University staff, Badlands National Park staff, and South Dakota Game Fish and Park staff. And with that, I will take any questions.
So it also looks like the different herds come into almost close contact with each other. And so that northern herd is in close proximity to this operation where they're bringing in new animals and stuff. So if this northern herd has this greater propensity for acquiring new stuff, do you know anything about the rate of transmission to like the other herds?
I don't know. And that operation, as this said, that operation, so every year the population just grows within, so they're not buying any new animals.
Okay.
They're just raising their own. Once they get to a certain number, they're selling off. So not coming and going with new animals. So we're hoping to test it to see if they actually have those certain pathogens, particularly mycoplasma, see if it's actually in that herd, and then see what our risk is there, and then go from there for management actions.
Is this the first time that you guys have looked at the prevalence of pneumonia with the Badlands?
Yes, yep.
Okay.
So this is the first time disease sampling's ever been done with respect to pneumonia.
And is it also the first time with the sheep? Because I know that, you know, Jenks has done stuff in the Black Hills, kind of similar to what you're doing now. Is this the first time you've included the herds nearby to see about transmission from the herds?
The domestic herd?
Mhm.
Yes, I believe it is.
With associated with the relocation or introduction programs, is there anything done, you know, veterinary medicine wise to address transmission? Or is it all left to purely what happens?
I guess I don't understand.
So I mean, veterinary medicine.
Yep.
You know, is anything applied, you know, are there any vaccinations, anything like that
okay,
done
that way. Yep.
to mitigate the spread of disease?
I think there is some vaccines that help domestic sheep and goats that will help them get over it or at least suppress the pathogen within them. But as for Bighorn sheep, there isn't.
Okay,
anything yet.
so you
Because-
basically rely on, you know,
Yep.
good veterinary care of the domestic herds.
Yeah, so as a
to
primary
reduce
management
transmission to
action is just keeping
wild
that
herds.
separation. And mycoplasma pneumonia, I should have said that too, because there's different strains of mycoplasma pneumonia. So you can create a vaccine for one strain,
Right.
and you can be exposed to a different strain, and there's multiple strains like this.
Any other questions? All right, one more round of applause. Thank you.